Okay, so the meiosis one has the prophase one. It is the longest and more complex compared to the prophase of mitosis, and based on the chromosomal behavior, it has 5 stages. First, liptotin, zygotin, pacatin, and diplotin, and then dikinesis in leptotin. The chromosomes become gradually visible under the light microscope. There is compaction of chromosomes that continues throughout the leptotin, and the second stage is zygotin. Now there is formation of synaptonomal complex. The process is synapsis, that is pairing of homologous chromosomes. It is synaptonomal complex, and that is a pair of synapse homologous chromosomes is bivalent or tetrad, because 4 chromatids are there. And but this stage is more clearly visible at pacatin. Now pacatin, it is the site for the crossing over that leads to recombination of genetic material. There is crossing between non-sister chromatids of homologous chromosomes, and the enzyme for this is recombinase. Okay, now crossing over is the exchange of genetic material between two homologous chromosomes. And then fourth stage is diplotin. Now beginning of diplotin recognized by dissolution of the synaptonomal complex formed during the zygotin, and the tendency of the recombinant homologous chromosomes of the bivalents to separate from each other except at the sites of crossing over, that is formation of the chiasmata in the diplotin. And another, just remember, in usads of some vertebrates diplotin can last for months or years. Diplotin stage in oocytes of some vertebrates. Now final stage of prophase 1 meiotic. Now prophase 1 is dikinesis. Now it is terminalization of chiasmata. Okay, now also by the end of the dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. Now dikinesis is transition to metaphase. Now metaphase 1, it is same bivalent chromosomes align at the equatorial plate. Okay, and the spindles fibers attached to the kinetic core of the homologous chromosomes. The anaphase 1, the homologous chromosomes separate while sister chromatids remain associated during their, so homologous chromosomes separate. And in anaphase 2, there is like splitting of the centromere, that is splitting of the sister chromatids. And this is it. Now telophase 1 is, the nuclear envelope, the nuclear membrane and nucleolus reappear. Okay, and cytokinesis follows, and this is called a dyad of cells. Okay, then there is interkinesis, which is a short-lived phase.